And this presentation comes from Hasibul Haque, and he's speaking on the molecular detection of Borrelia in human tissue. As a brief introduction, MD Hasibul Haque, and I'm butchering your last name, I'm so sorry, is a master's candidate at Mount Allison University doing research on the molecular diagnosis of Borrelia in human tissue. He received his Doctor of Veterinary Medicine, DVM, as well as Masters of Science in Parasitology degrees from the University of, again, I'm going to butcher it, Russia and Bangladesh Agricultural University, respectively. His future plan is to get a PhD in the field of molecular biology. Please join me in welcoming Hasibul.
So hi everyone, my name is Hasibul Haque, and today I'm going to talk about molecular detection of Borrelia in human tissue. So as you already know that Borrelia is the Lyme causes bacteria, so I'll talk about it later and more. So before we move forward, let's talk about a little bit on my presentation outline. At first, I will give a brief discussion on the Borrelia, and then I will talk about my research on testing of tissues for Borrelia infection. So, as already Joey said a little bit about Borrelia, so I'm going to more about it. So, Borrelia is a spirochete bacteria, which means it has a corkscrew shape. We can see it, and it has endoflagella, which actually go throughout the body and helps it to dig to the tissue and swim in the blood. So there are 52 known species of Borrelia. Among of them, 21 are members of the Lyme disease group, and 29 belongs to the relapsing fever group. And Borrelia can take other forms of under unfavorable condition, and they can form blebs, and also persister or round bodies, and biofilm, in a suspension, and mostly in the culture. So what happens, as one bacteria has different form, it actually creates a problem for the physician to treat or the bacteria with antibiotic therapy. And there are certain forms like persisters or round bodies that are highly resistant to antibiotic. And sometimes these things actually challenge the current antibiotic treatment protocol throughout the world for Lyme disease. So why molecular detection for Borrelia in human tissue is important? Actually, molecular methods enable direct detection of pathogen at the onset of the clinical signs. It serves as a method for identification and typing of Borrelia in the specimens or cultured isolates. And it also enables detection of Borrelia and co-infection with different Borrelia species in a particular sample. Now, for my research, I hypothesized that molecular examination of biopsy, necropsy, and autopsy tissue for Borrelia and other associated bacteria will allow us to understand the relation of Borrelia infection to the associated disease. And from this hypothesis, I think that it leads me to evaluate the value of direct molecular detection of Borrelia in human tissue. Now, how I get my sample? For testing of the human tissue, I get samples from 3 sources, basically. They are from autopsy, biopsy, and necropsy sources. And during the collection of samples, targeted tissues actually synovial membrane, major vasculature, nervous tissues. So for autopsy and necropsy, I get 2 types of samples. One group is 95% ethanol submerged tissues, and other group is paraffin embedded tissues. So for ethanol submerged tissues, I do nested PCR after extraction of DNA. And for paraffin embedded tissues, I do molecular beacon immunostaining, and hopefully in the future, I'll do silver staining too. And for biopsy, at first we do culture, and then extract DNA from the culture and do our nested PCR for Borrelia. Right now, in my project, I'm doing 3 tests for Borrelia, and these are nested PCR, molecular beacon, and immunostaining. So from the next couple of slides, I'll talk about these techniques one by one. So at the beginning, let's talk about the nested PCR. It's a sensitive technique for amplifying low abundance of DNA, and in a special form of PCR from the normal PCR. Here, in the nested PCR, we use 2 different primer. One is outer and one is inner. So from our first round product, we use a PCR product to do a second round of PCR. And this second round of PCR goes for gel run. So now I'm going to have a brief description how I do this nested PCR thing. So sample preparation for nested PCR actually implemented inside a PCR station. After preparing the sample, we put them on the thermocycler, and it takes around 2 hours to complete the whole cycle. And then we load our PCR products into a gel. And after gel run, we found an image like this. So next, molecular beacon. So in molecular beacon, we use a single standard oligonucleotide hybridized probe. So in this probe, in one end there is a quencher, and the other end there is a dye. So when this probe is not hybridized, it forms a stem loop structure and binds with this quencher quench the dye and prevents it to emit fluorescence. Now, when hybridization occurs, these probes attach with the target DNA. So quencher and dye separated from each other, and it starts to fluorescence. And in that case, it looks like this. You can see that these are the fluorescence. For immunostaining, we use antibody, which is conjugated with HRP, and it actually binds with the Borrelia. So after this step, we do a wash, where unbound antibody washed off, and then we add a TMB. So HRP attaches with the antibody, reacts with the TMB, and produce a colored precipitase. And under microscope, we found an image like this one. So here, I will talk about a case study where I tested all these 3 molecular tests. And the patient was a 70-year Caucasian male. He had a history of Lyme disease, and which was confirmed by 2-tier Canadian serology. Received standard treatment of antibiotics. And during the time of death, his cause of death was severe coronary artery disease with associated extensive atherosclerosis. So from this patient, we found 2 types of tissues. One type was ethanol submerged tissue, and other one is paraffin-embedded tissue. So the ethanol submerged tissues samples from different body parts were tested by nested PCR and found positive for flagellin B. And later on, we confirmed it by sequencing. And here, each band shows different tissues from the same patient. After completing a molecular beacon, you can see that the Borrelia is glowing in that area for the BB3 sample. And in the left, we have a positive control from mouse brain, which is also infected with Borrelia and also emitting those fluorescence And in the right, there is a negative control of mouse brain which does not have any glowing Borrelia. In the immunostaining of BB3, we use the same paraffin-embedded tissue sample. And for the BB3, we found these are different forms of Borrelia. Like you can see, this is brown body persister, and also like a spirochetal form. And in the left, the positive control, we found the same thing, like spirochetal form, and also persisters or brown bodies. And as usual, in the right, there is no staining. So to conclude for BB3, we can say that Borrelia found in all targeted tissue samples, and molecular beacons show round bodies and spirochetal forms. Immunostaining also shows Borrelia in different forms. In this case, standard treatment didn't eliminate the Borrelia. So as a whole, we can say that these results highlight the value of direct molecular detection of Borrelia in human tissue. In this case, the molecular findings are completely consistent with tick exposure and serology. In other cases, however, this information may not be known, and molecular detection offers the possibility of determining if Borrelia is present. This approach also offers the opportunity to address other research questions such as Borrelia tissue distribution and correlation with tissue damage. So I have some future plan with my samples. I will do some silver stainings, and for this I have to optimize the protocol. And I have some more samples for processing, and I'll develop some new primers for other associated disease. At the end, I would like to express my gratitude to my supervisor, Dr. Vetloyd, for her guidance, encouragement, and useful critiques in this research work. I would like to thank Yvonne Mosley for arranging the tissue donation, Julie Lewis, and my lab mates for their cooperation and encouragement throughout my study. I'd also like to thank all of my funding organizations, NBIF, NSARC, and CanLine. So these are my references. Thank you. If you have any questions.